In this lecture, I will discuss about TH17 and iTreg cells, how these cells are generated and what are the effector functions of these cells. What happens if there is any infections from this bacteria and fungi, then our immune cells and epithelial cells can secrete some chemicals that are called pro-inflammatory cytokines. Example of these cytokines are IL-1, IL-6. For the generation of TH17 cells, other two important chemicals are IL-23 and TGF-beta. And among them, this TGF-beta and IL-6 is very important for the generation of TH17 cells. So what happens? Naive T helper cells, when receive this cytokine, the receptor, these T helper cells become TH17 cells. And when they receive, the cells receive these cytokines, they start to express a master regulator. In case of TH17, the master regulator is Rod gamma T. Likewise, the TH1, TH2 cells, these cells also inhibit other cell production like TH1, TH2, inhibit other cell production. And when this Rod gamma T is actually produced in TH17 cells, these cells actually secrete some cytokines like IL-17. From this chemical cytokine IL-17, it gets its name TH17. And another important is IL-22. These cytokines actually attract neutrophils. This is very important. So they attract this neutrophil. So these TH17 cells, actually main function of these cells is to actually induce inflammation. And in response to these cytokines, they recruit neutrophils so that neutrophils can kill this bacteria and fungi. So this is the generation and its major effector functions. The next cell is T regulatory cells. I means induced. So this is actually an induced T regulatory cell. But in our body, in our immune system, there is another type of T regulatory cell that is called NT regulatory cell. This N means natural. So this T regulatory cell is natural. In our body, these nTregs are produced from the thymus. So they are actually natural. But these cells, T regulatory cell is produced in the periphery, not from the thymus. The main function of these T regulatory cells, I am talking about this NT regulatory cell, to suppress immunity or immune cells. What happens? If there is any infections, some cells are present. Some cells like TH1, TH2, TH17, cytotoxic T cells, these cells actually try to kill the pathogen. Okay, so when there is any infections, the number of these cells increase very fast. But after antigen elimination or the pathogen eliminations, the number of these cells must be decreased. This is called homeostasis, because our immune system try to maintain a balance. Okay, when there is infection, the number of these cells should be high. But when the pathogen is actually eliminated or antigen is eliminated, the number of these cells should be low, as it was in the previous conditions before the infections. So this function, this lowering of this number, is performed by NT regulatory cells. So this T regulatory cell is very important for autoimmunity. If the number or if this NT regulatory cell is actually non-functional, then autoimmunity is actually generated. So these cells actually suppress these immune cells so that a homeostasis, so main function of this cell is to homeostasis. Homeostasis can be maintained in our immune system. Homeostasis means simply balance. But what happens? How the T regulatory cell is generated? There is a reason. And the reason is tumor. The reason is tumor cells, or you can say tumor mass, specifically breast tumor. What we have seen, that this tumor cell try to escape our immune system and they manipulate our immune system so that immune cells do not kill this tumor. These tumor cells can be killed by cytotoxic T cells. So cytotoxic T cells are present in the vicinity of this tumor, and the tumor cells try to survive. They try to escape from this killing. So what happens? Specifically, we have seen that breast tumor secrete huge amount of a chemical called TGF-beta, and this cytotoxic T cells, to increase its number, it secretes another molecule called IL-2. So all these cells are secreting IL-2 cytokine for their own proliferation. It is for their own benefit. So IL-2 is for their own benefit, and TGF-beta is producing from the tumor. And what happens if there is any T helper cell come across this tumor? They receive this TGF-beta, and when they receive this TGF-beta, this T helper cell starts to produce a master regulatory that is called FOXP3. FOXP3. This is the master regulator factor of actually transcription factor that regulate IT regulatory cells. When FOXP3 starts to express in this T helper cells, this cell actually expresses a very important molecule. I have already discussed about this molecule in T cell activation. This is called CD25. Huge amount of CD25 is produced in these cells than the T helper cell. If there is 5 CD25 as an example, if there is 5 CD25 present in one cytotoxic T cells, 5000 CD25 can be present in the FOXP3 positive cells. So they express many CD25 molecules in their surface, and what happens? These cells actually deprive these cytotoxic T cells from their production of this IL-2. They are producing, these cells are producing IL-2, but this IL-2 actually received by this molecule, because there are many receptors present on these T helper cells that is expressing FOXP3, because FOXP3 help to produce or transcribe CD25 molecule. And now the cytotoxic cells actually deprive. So this means the cells actually deprive other cells by absorbing IL-2 molecule, because they are expressing CD25. This is the IL-2 receptor. CD25 is IL-2 receptor. I have talked about this is in my T cell activation class. This is actually alpha chain of the IL-2 receptor. When these cells, these FOXP3 cells receiving TGF-beta and IL-2, it becomes IT regulatory cell. As the NT regulatory cell can secrete suppressive molecules like IL-10 and TGF-beta, these cells also can secrete these suppressive cytokines. These are the suppressive cytokines, and you can see this TGF-beta also amplify the production of these FOXP3 cells. So this is actually a mechanism of immune evasion. And in 2018, two scientists, eminent scientists, J.P. Ellison and Professor Takasu Honjo, they rewarded Nobel Prize for the discovery of monoclonal antibody against very important two molecules that is actually expressed by these T regulatory cells. One is CTLA4 and PD1. They discovered a monoclonal antibody against the CTLA4 and PD1. CTLA4 by J.P. Ellison and PD1 by Professor Honjo. And this is actually the function of this IT regulatory cell. This is called contact dependent inhibitions, because this cytokine can be secreted and it can function indistantly. So this is contact independent. But this IT regulatory cell also express a molecule that is called CTLA4. This molecule can inhibit if there is any contact between these cells. So this IT regulatory cell can inhibit any cells that contact with these cells, like dendritic cell, macrophage, like antigen-presenting cells. In naive T cells, there is a co-stimulatory molecule called CD28 that can bind with CD80 or CD86, that is co-stimulatory molecule, and this is a receptor, co-stimulatory receptor. So when this molecule bind with CD80 and 86, second signaling occurs. This is very important for the activation of T cells. But do you know that this CTLA4, this inhibitory molecule, also bind with CD80 and CD86 molecules, same molecule that actually bind with the CD28, and that is very important for the T cell activations. So this CTLA4 can bind with this same molecule and with higher affinity. Not only bind, it bind with this molecule with higher affinity, much more higher affinity than the CD28. So what happens? If the IT regulatory cell starts to express CTLA4 molecule, then it engage CD80 and 86 molecules with high affinity, so that no CD28 molecule of T helper cell, naive T helper cells can engage with the CD80 and 86. So they do not get the co-stimulatory molecule, that is the second signal for the T cell activations. The first signal is TCR and MSC molecule interaction. The second signal is the CD28 and 86 co-stimulatory molecule signaling. So these connections will not occur if these IT regulatory cells, let's assume this is the IT regulatory cell. So at first it can secrete IL-10 and TGF-beta. This is contact independent inhibitions. It can secrete IL-10 and TGF-beta, and this IL-10 TGF-beta can reach many distant of immune cells and can suppress. But contact dependent mechanism is also present, that is called contact dependent. So there is contact independent, that is by secreting IL-10 and TGF-beta, and contact dependent by CTLA4. So there are two inhibitory mechanism of IT regulatory cells. So what happens if this IT regulatory cells is expressing CTLA4, and let's assume this is a dendritic cell, this is a dendritic cell that is expressing CD80, this is CD80 or CD86, then the CD80 and CD86 bind with the CTLA4 with high affinity. So all the CD86 actually engage with CTLA4 and no CD86 actually remain or unengaged for the CD28 engagement. So let's assume this is the T helper cell. So T helper cell come, it will recognizing with the dendritic cell MHC2 molecule, but this T helper cells expressing CD28, but it cannot bind with the CD86 because there is CTLA4. So when iTreg cells bind with this dendritic cell with this CTLA4, this T helper cell cannot bind with the same CD8 and 86 because the same ligand, because the CTLA4 have high affinity. And not only that, this binding, when the CTLA4 and CD86 bind, they inhibit these dendritic cells to perform its antigen presenting functions. This is very important. This is called extrinsic function. There are two functions of the CTLA4 I am not mentioning here. One is called cell CTLA4 intrinsic actually inhibitions, and another is extrinsic. So inhibition of this dendritic cell by the IT regulatory cell of the CTLA4 is called extrinsic inhibition mechanism. So this CTLA4 can inhibit the dendritic cell so that it now can not represent antigen presenting as a antigen presenting cells or antigen. So this T helper cell do not get any help from these dendritic cells that engage with the IT regulatory cells.